So my name is Wan Fen. I am from Department of Medical Microbiology from Universiti Malaya in Malaysia. So let me just take 1 minute to introduce our institute, Universiti Malaya. Some of you might not know that Universiti Malaya and US were once one single institute in the past in 1905. So, and then we were splitted later on, just like the fate of Malaysia and Singapore, we've splitted into 2 countries. So that photo shows our campus. It's a pretty green and beautiful campus. You're welcome to visit us if you have a chance. So we have around 3,000 undergraduate students and around 2,000 academic staffs in our institute. So the one below is our Universiti Malaya Medical Center together with the Faculty of Medicine where we work in. So let me start with just with a quote in the law of conservation of energy, which says that power cannot be created or destroyed, but only can be transformed. So in our world, we receive the energy from the sun. So we get the solar power, which transform into electrical power that move our car or light our light and so on. So if the solar power go to the plant through the photosynthesis, it's transformed into chemical energy. So when humans consume the plant, we get energy into us. So why we are saying about this? Because the energy is really important for us, especially in the case of immunology. So mitochondria is mitochondria is our powerhouse in the cell. So how we generate energy in us is through the electron transport chain, and glycolysis as well. But through the electron transport chain is a more effective way of how we generate the energy. So when the electron is transported, so hydrogen will be pumped into the intermembrane space. So, and this eventually build out a gradient inside the intermembrane space called as the proton motive force. So if you imagine this, like in the case of this mitochondria, so a lot of proton build up in this intermembrane space, and there's no proton down here. So it build up a force just like a hydropower dam, in which when you store the water up here, and once it flushes down, it creates a very strong power which spins the turbine, which is the ATP synthase, which can eventually create the ATP. So this power generation is especially important in T cell activation because in the case of naive T cells, we know that naive T cells is very small and cute cells in which it has a small cytoplasm. It doesn't consume much energy because we just take what is needed for normal homeostasis and also the patrolling. So, but once the naive T cells is exposed to its connect antigen, it will undergo a clonal expansion from 1 cell into thousands of cells, and it has to undergo its killing activities and release a lot of cytokines. So that is a huge dramatic change in the power demand. So from this process to this process, so what happened here is the naive T cells is using mainly oxidative phosphorylation for power generation. But once it turns into effector cells, it acquires what is known as aerobic glycolysis, or similar to the case of Warburg effect in the cancer cells, where a huge energy is needed. So aerobic glycolysis is being utilized. So upon clearance, the cells eventually become memory T cells, which utilize the fatty acid oxidation. So in the case of chronic antigen presence, like in the case of HBV or in the case of HIV, what happened is that the CD8 positive T cells, because of long-term exposure to to the antigen, it eventually become exhausted. So we are interested to look into the metabolic program in the case of exhausted CD8 T cells. So what happened here is we did a T cells, CD8 positive T cells stimulation every 2 days. So every 2 days, and what we can see here is that activation marker can be increased, but it wouldn't last for long. So it eventually comes down after day 10. In contrast, the PD-1 will eventually increase, marking the exhausted T cells. So the cytokines that increase also eventually come down. So this is also why we cannot work continuously for 10 days. So we need to work for 5 days and take a break during the weekends. So before we continue working to continue to get activated. So when we look at the mitochondria during this activation phase, what happens is that the healthy mitochondria is increased upon activation, but it quickly reduces following the continuous activation. So what happened simultaneously is that the dysfunctional mitochondria has increased. So this means that the mitochondria function is being impaired in the case of exhausted T cells. So looking into the context of HIV, so we know well that HIV continued to be a huge problem to us, with over 39 million people still living with HIV. So it is well known that HIV targets the CD4 positive T cells and kills the cells. And as the CD4 positive T cell number comes down, the viral titer will increase and eventually leads to opportunistic infection and also AIDS. So nowadays we have the heart treatment, which has improved dramatically. A lot of patients by reducing the viral replication. So most of these patients that are responding well, we call them as treatment responder, but there are still a subset of individuals which do not respond, and we call them as treatment failure. So when we look at their CD8 positive T cells and compare between treatment responder, treatment failure, and treatment naive population, you can see that the CD8 T cells is much lower in treatment failure and treatment naive population compared to treatment responder. The treatment responder has a very low or non-detectable viral load, whereas the treatment failure and treatment naive has a high viral loads. So with the increase of viral load, they have a lower amount of CD8 positive T cells. So the reason why of the decrease of CD8 positive T cells is because of the T cell exhaustion that eventually causes the cells to die. So when we examine the T naive in each of these patient cohort, you can see that the naive T cells and effector T cells are much lower in treatment failure and treatment naive population, whereas in contrast, the terminal differentiated T cells as well as exhausted T cells are increased. So in terms of their ability to produce the cytokines, so the environment or treatment responder that can produce much higher interferon as well as TNF in response to stimulation compared to those viremic patients from the treatment failure patients. So when we examine that granzyme B, perforin, granulysins, and cytokines, you can see that those effector cells has a high expression of these enzymes, but compared to the effectors, compared to the exhausted cells, which lost the activity. So when we look at the mitochondria in treatment responder, treatment failure versus the treatment naive, can see that the percentage of healthy mitochondria decreased, whereas the percentage of dysfunctional mitochondria has increased, which suggests the mitochondrial impairment in these patients. So we also look at the glucose receptors among these patients, and treatment responder has a lower or moderate expression of glucose receptors, whereas treatment responder and treatment naive, they have increased percentage of GLUT1 high T cells. So we further did a seahorse analysis to check their mitochondrial activity. So this is a seahorse mitostress activity in which different medicines different drugs are being introduced. Like for example, FCCP caused the flux of proton into the matrix area and enables us to check the maximal potential of the mitochondria, whereas retinol and antimycin block complex 1 and 3, oligomycin block the ATP synthase. So this is the seahorse result. So we compare between the treatment responder and treatment failure patients. Treatment failure patients, which are viremic, their basal level of the OCAR, or oxygen consumption rate, is much lower compared to the treatment responder. So when different drugs are being introduced, like for example, the FCCP is introduced, we want to check the maximal potential of ATP being generated, but we can see that TR is responding to the drug, but treatment failure cohort is not responding to the drug, marking that their mitochondria activity is impaired. So when we check the basal activity, maximal activity, as well as the ATP, all of these are being reduced in the treatment failure compared to treatment responder. So this chart shows the ECAR, or extracellular oxidation rate, which tells us the glycolysis activity. We can see that not only that the mitochondria activities are being impaired, the glycolysis activity is also impaired in TF compared to the TR. So ATP link ATP link aspiration is significantly reduced, which suggests the overall energy are being downregulated. So we also look at the transcriptomic analysis of this, in this case, the viremic patients, and we sorted the cells into naive, early, intermediate, late effector cells with PD-1, and also exhausted T cells. So we use a nanostring encounter, which the panel contains 700 genes of metabolic pathway. If we can see from this heat map that a lot of metabolic genes in the exhausted T cells are being downregulated. So it has a much similar pattern to naive T cells compared to other subsets. So a volcano plot shows that most of this gene in metabolic pathway are downregulated. There's hardly any genes that are being upregulated. So a closer look into the TCA cycle genes, like for example IDH1 and so on, they are similarly being downregulated in the exhausted T cells. They are high in the effector T cells, but they are low in the exhausted T cells. So the real question is really since the CD8 T cells, when it's exhausted, it has impaired OX4 and glycolysis. Is there a way we can intervene the mitochondria and to repair the mitochondrial respiration and thereby reinvigorate the exhausted T cells to enable it to come back to reactivate and fight the HIV again. So similar to the concept in the case of cancer immunotherapy, where people use anti-PD-1 against the inactivated T cells. So this is one of the work that we have done, in which we collected the cells from different patients, and we put in the antioxidants which targets the mitochondria, and we can see that upon treatment, the healthy mitochondria increase, whereas dysfunctional mitochondria decrease. But however, when we examine the PD-1 cytolytic T cells or glucose receptors, there's no significant changes following the treatment. Same goes to the cytokines production. There's no recovery as expected following the drug treatment, despite increase in the healthy mitochondria. So they come to our conclusion that so treatment responder and treatment failure, they have a very different CD8 T cells percentage. So those patients with high viremic, they are unable to suppress the virus HIV because their CD8 T cells are being exhausted. So effector T cells, it works well with healthy mitochondria, whereas exhausted T cells, despite having more glucose receptor, the glycolysis is impaired, so as the mitochondria being depolarized and dysfunctional. So, and a single or dual target therapeutic strategy may not be insufficient. So in the future, we're thinking of doing a more general intervention on how we can revive the exhausted CD8 T cells. So most of this work are being done by a student, Yiting. She's now work as a postdoc fellow in Jiwa's lab in NUS. And some of the work are being done by Carmen, who's in the audience, together with Chun Hao and Zhiying. So I also like to thank Prof. Adiba for supporting the work and some of the clinicians who helped to recruit the samples. Okay, with that, I thank you.
So do you have any suggestions then how to recover mitochondrial function in exhausted T cells?
Because we're seeing that the coupling effect is not affected, the mitochondria itself, although they are unable to produce the energy, but it is not just the mitochondria that is affected, but glycolysis is also affected. So it looks more like a global suppression of the whole activity of the T cells rather than a single pathway alone. So that make the work a little bit challenging.
Sure.
So I guess a related question. What we see in our models is that the more inflammation you have, then the more likely the T cells are going to move into an exhausted type phenotype. So I guess one of the questions would be use of your anti-inflammatories to earlier on before they transition to see if that will prevent them becoming exhausted.
Anti-inflammatory. So anti-inflammatory and antiviral are two different pathways, right? Some people did suggest that if you want to have more for the T cells to divert into antiviral path, you got to suppress the other side, which is the anti-inflammatory. Yeah. So I guess that is a good suggestion that maybe we can look into it.
Well, you've got a great model, I guess, to test it.
Thank you. Yeah.
Thank you.